Well, thanks for that introduction, and thanks to the organisers for the opportunity to give you an update on AMPLE. AMPLE is our pipeline for unconventional molecular replacement. So maybe if your target is a completely novel fold not represented in the PDB, or you can find a homologue in the PDB but it's a bit too distant from your target to work easily, AMPLE has some options that you might want to look at. So this talk is going to be in 3 little chapters. The first is going to be a little bit historical, looking at the first incarnation of AMPLE. So there'll be no new data in that part, but that explains how AMPLE works and some of the methodology that we eventually applied in different situations as well. I will also explain in that section how to use AMPLE, the different ways in which it's accessible to you. In the second part, we're looking at experimental structures, mainly crystal structures, so distant homologues that don't work easily by molecular replacement, and a couple of options in AMPLE to work with them to extract the maximum value of homologues you can find in the PDB. And then the third part is going to be about the big excitement in structural bioinformatics recently, this evolutionary covariance, allowing you to predict residue contacts, and one of the many ramifications of that new development. So this talk fits quite nicely into the topic of this meeting, the integrative and rational molecular replacement, because AMPLE is all about integrating bioinformatics developments and making them useful, translating them to structural biology so that useful to you as a crystallographer. So there's going to be 3 slides introducing the 3 little sections and explaining a bit of the bioinformatics behind the 3 ideas. So the first version of AMPLE was built to work with ab initio models. So ab initio modeling is what you can try if you can't do your regular homology modeling, because there's nothing similar in the PDB. You've got a novel fold, you want to predict it, you're in an ab initio modeling situation. Over a couple of decades, software like Rosetta and Quark has got quite good at this, and the method they use, it's interesting to look back briefly, is fragment assembly. So people realized quite early on that proteins, of course, are built from repeating building blocks, which means that for any given structure, you can actually reconstruct it quite well using chunks of unrelated proteins. And that opens up the possibility of ab initio modeling if you can find suitable fragments, if you can link them together. And this animation is showing you a Monte Carlo kind of system, which is generally how these folding pathways work. And then the last figure is explaining how you identify the likely best predictions in your set, because this modeling is quite difficult. You do it hundreds or thousands of times, so you get a big pool of candidates, which are often quite diverse. How do you pick out the best of them? Well, the best way turns out to be to cluster them and find groups of them that are similar. So if you make 1,000 models and 3 of them turn out broadly similar, that's your likely candidate fold prediction, and this is representing the identification through clustering. And clustering is also the first step along the route to producing ensemble search models for molecular replacement, and that, as you'll hear over and over, is very advantageous. The second section has a couple of ideas, but one of them is based on non-trivial editing of single distant homologues, so structures that you can recognize homologous to your target but which are not close enough to work easily with the usual kind of editing and tweaks. And one route to make the most of those targets is based on this recent finding that flexible regions evolve more quickly than rigid ones, and that means that we can use rigidity and flexibility as proxies for parts of the structure that are likely to be evolutionarily conserved or evolutionary variable. And you can see the correlation between substitution rate, the degree to which residues evolve, and their rigidity here. So we can predict roughly which parts of a protein are going to be variable in evolutionary terms. And of course, that means we can narrow down, we can chop down a protein structure to reveal the part that's likely to be better conserved across a family. And that, of course, is likely to be the bit that's most, that's going to be found in your unknown target as well. So we can do that in a single structure, but we can also make ensembles using this software. I'll show you that later on. And in these ensembles, we're visualizing the flexibility. So this red region, the tail, is very flexible, and we can see that this derives from a single structure, and we can use editing to get rid of these variable bits and leave the conserved core, which is likely to be what's most useful to us. And then the third part, like I say, is to do with evolutionary covariance, which allows us to predict residue contacts. This is some of the software that's prominent in the area. The basic idea here is very simple, which is that if you have a biological contact between two residues, which is very important, and that can be within a molecule for folding it or between two molecules for the biological function, then natural selection may only allow one to evolve if the other one simultaneously changes. So maybe one's allowed to get bigger, but only if the second residue touching it gets smaller at the same time. So there's a kind of compensation. So what this leads to is covariance between positions, and we can recognize that with the appropriate statistics in large sequence alignments. This is the direct coupling analysis, these days often complemented with some kind of deep learning to get the best results. But what comes out of that is a prediction for your protein based on this analysis of the homologs about which residues are touching which other residues. So this is extra information about the protein structure, which actually has big ramifications across structural biology when you stop and think about it. But here is particularly useful because it allows us to build better ab initio models with that extra information factored in. And that makes a difference to molecular replacement. So the first little section, just explaining how AMPL works and some early-ish results that we got from it. On the left are these fragment-based ab initio models, a large number of them, like I say, with software like Rosetta and Quark. The keywords here on this slide are picked out in red, so we're clustering things, we're truncating things, we're editing them down, and the idea is to make ensemble search models, which we're going to trial by molecular replacement. So like I say, among this large set of models, we say the most likely are the ones that are forming large clusters of similar models. So here's a large cluster found within these results. Very importantly, within that cluster of similar structures, it's known, we and others realize, that the variable parts are likely to be less reliable. And so we can progressively eliminate those and hopefully leave ourselves with something smaller, still suitable for molecular replacement, but more accurate in terms of its resemblance to the unknown target. So we take the cluster, we do this progressive truncation at 20 different thresholds. We use a program called Theseus, which is a maximum likelihood structure superposition, which best superimposes core regions. Then we compose search models at different subclustering thresholds, so quite 1, 2, 3 angstrom thresholds. Some of these details are not that important. And then initially we had these 3 modes of side chain treatments, although more recently, as you'll see, we've reverted to looking at polyalanine only. But from each of these clusters, we can generate up to 180 ensemble search models. Often the first cluster is where the action is. In very hard cases, you may need to explore other clusters in exactly the same way. So we have a large number of search models, and that's really the original bit of AMPL. All of the rest that comes after that is other people's clever software. We have to acknowledge that. So Mr. Bump is what we use to process them. Phaser, or optionally MolRep, places them. And ShellXC turns out to be very important because, as you'll no doubt hear elsewhere as well, it can give you resolution allowing very good, reliable statistics that tell you yes or no whether your molecular replacement solution is correct. And that can be hard to know otherwise, absolutely. I'll show you some examples of that. And then you can also ask AMPL to trace further rebuild those ShellXC traces with Buccaneer or ARP-WARP. And I'll come back to the importance of citing the underlying software later on. So just going briefly through some early results. So the time we were doing this ab initio modeling was only really for small proteins, so we were quite conservative. We went up to 120 residues. The red and the yellow are the successes, the blues are the failures. So we solved about 43% of quite a large test set. We realized, of course, that the larger proteins were more difficult because it's harder to model larger proteins. We also realized that alpha proteins work better than beta proteins. And again, that's largely because the modeling does a better job of alpha proteins than beta proteins. Just to show you one example of the kind of ample rationale, this is the target that we solved. Ample produced a cluster that looked like this. So again, you can see this red part is quite consistent between those structure predictions. That's a sign that this is reliably predicted. The green and the blue is more variable. That's a sign it's less reliable. So one of the truncations that Ample produced looks like this. That was enough to solve the structure with the benefit of those side chains, and this is what ShellXC rebuilt. So Ample does that progression progressively, so you get quite complete search models and very fragmentary ones, and we've realized that you can get successes in all of these categories. So here the modeling was relatively reliable for the whole fold, and that worked. Here it was about half of the target, and that worked. And here a very small fragment was placed sufficiently accurately by phaser to allow ShellXC to trace out the whole structure. So one of the benefits of Ample is you're sampling all of these different strategies simultaneously, really. You've got some money betting that your fold is right and some money betting that some small chunk will get it by good fortune. Just to mention coiled coils, they can still be awkward. You heard an example of that in the MR clinic. Ample does a pretty good job of coiled coils, and so does Archambault. A more recent paper shows that. What was nice with Ample was we realized that you didn't need to predict, you didn't need to know anything about the coil. Coiled coils come in a variety of morphologies, as you can see, and Ample seemed to solve them all equally well. But actually you can do a better job if you do know something about your target, and Owen Davis is going to show some results for that. He's going to show that if you know that your coiled coil is a dimer, then you can model a dimer and use an oligomeric search model from Rosetta in Ample. This is an example of that, and I only show it to make one general point, which is that like here, we're trying to solve the purple structure. The Rosetta model in this case is not very good, but we can trim it down to something that does represent part of the model well. And in particular, you'd notice the curvature of this helix. So Ample will often capture small local features in an authentic way, even if the ab initio modeling hasn't got the global fold exactly right. And that's absolutely fine. We also looked at transmembrane proteins. Quite a few of them solved, again, over a range of sizes. We introduced a very simplistic kind of ideal helix mode just to convince ourselves that we were doing more than producing search models from Rosetta, which were basically helices. For the transmembrane ones, the ideal helices, even our simplistic implementation works surprisingly well, but these two cases here, these were the lower resolution cases, these actually needed explicit modeling to solve as well. So how can you try Ample? The very easiest way is to use the Quark server. So Quark is a fragment-based ab initio modeling protocol very much like Rosetta. They run a server which will give you your results back in 12 to 24 hours. It accepts sequences up to 200 residues. And you can send the result of that, which is one single file, to CCP4 online, and Ample will run for you there. If you need more flexibility, you can install Rosetta, you can take fragment files from a different server, and then you've got the two GUIs. Don't know why I've lost that. You've got the command line, and you've also got CCP4 Cloud coming, which is where we hope to implement relatively complex options in a very accessible way to users in the near future. At the command line, it can be as simple as this. Here's your Quark models, your sequence and your reflections. If you're modeling, it's a little bit more complicated. You need to give it the fragment files, which are the raw material for the modeling, and you need to tell it where Rosetta is. You've got an extra flag if your target is transmembrane. And if you want to try this simplistic ideal helix mode, you can. However you run it, you'll get an ample log file. This may be presented more elegantly in a GUI, but this is just the command line version. Just to pick out one or two things here. This list of ensemble names, the code here says that this is coming from cluster 1. It's truncated down until 42% of the residues are left. Radius 3 of that subclustering, and polyalanine. In this case, the phasor statistics were already very good, but you can, where the resolution allows, ShellXC is very good at distinguishing between successes and failures. So here are some unambiguous successes with ShellXC correlation coefficients above 25, long average chain lengths, and then there's a very nice clear gap between ones that haven't worked. And in this case, the ample run finished early because it found some successes. And where ample thinks that ShellXC has worked, you can ask it, like I say, to rebuild further in Buccaneer. In this case, that got down to our freeze of about 35%. So, just have a quick drink. Next little section is about ideas we had of adapting some of these ideas to work with experimental structures. So, and this is really about repurposing that whole truncation. So what you've seen so far is that with ab initio models, those variable parts are less reliable. We can truncate those off and leave ourselves a nice, more reliably modeled core. We can apply the same kind of truncation procedure to different sets of structures. So it turns out it works very well with NMR ensembles. I won't show you that. That's the reference there. If you have several distant homologs, you can superimpose them and use the truncate off the variable part to find the conserved core that's likely to be in your target. And like I said, there's this interesting correlation between packing and evolutionary conservation, which we can exploit and produce ensembles even from a single distant homolog, and I'll show you how we do that. So, but just to visualize the situation you might be in, you might have one of these is your target, the other one is the homolog from the PDB. You can recognize the evolution relationship between the two, but of course they're very different, and no amount of manual fiddling and tweaking of loops is going to make this structure solve with something derived from this. So how do we identify this conserved core, which is actually shared between them? Although the overall structures are very different, they do share a conserved core, and this would be very suitable to solve either one of them. Well, if you have multiple structures, you superimpose them. I'll show you that. And it turns out that making an ensemble from a single structure and truncating back the variable parts, which are the flexible parts, which are the evolutionarily less conserved parts, is the best way to exploit a single structure, or a good way to exploit a single structure. So with several structures, ample will start from what's shared between the three. Here we've got three members of a big superfamily, and we're going to try and solve a few others in the family. So you can see these are the three. They're very diverse. This is what they have in common. And if you watch this animation, ample will truncate down progressively, producing search models of all of these sizes. And the smallest ones contain, indeed, this region at the end of this beta sheet, which is exactly where the conserved catalytic site is. So the superposition is working and the truncation is working to zoom in on that shared catalytic core. And to cut a long story short, it does a better job processing them this way than in Mr. Bump's regular superposition algorithm. These numbers are numbers of successful and total numbers of search models produced for each of these runs. So if you only have a single structure, then you have to be a bit more creative. The idea here is to exploit that relationship, that the less well-packed parts are going to be less conserved, and so they're likely to be less useful in the phasing. So we can take a single structure here, we can generate this ensemble. This is a program called Concord. It's a kind of distance geometry way of taking a structure, looking at the contacts between residues, allowing them to vary slightly and generating a family of structures in the vicinity of your starting structure. And in that family, regions that are not well packed become very variable, and this conserved catalytic site here is very rigid, and that remains right to the end. So we truncate off the variable bits. They're likely to be less useful, and we focus in on the shared conserved catalytic site at the end of this strand here. So that's what we'd already done and published last year. We looked at this method here, which we came across at a conference. Concord is not slow, but it can take up to half an hour on a single core for a larger protein, whereas this, which is a normal modes-based method, is very, very quick. Of course, normal modes is not new to molecular replacement, but before, what people have done is said, okay, I'll try some of the extremes of this normal mode. Here's my structure. Normal mode 1 takes it here or here. I'll try these two structures as search models. What we're doing is taking the top 10 normal modes and positions along them, superimposing them. And again, this is a bit more messy, but again, the less well-packed regions come out diverse, and the well-packed catalytic site remains conserved. So we can truncate down those ensembles derived from a single structure and try and solve this set of targets, which are genuinely quite distantly related. You can see the percentage identity there. Particularly these two at the bottom are in a whole different area of the superfamily. They're very difficult. So Mr. Bump, in a kind of comparison with some structures that were edited down initially by an expert crystallographer and then processed further by Mr. Bump manages to solve 1, whereas Concord solves 6, and this NOLB, which is this normal modes program, solves 5. So each of these represents quite a big improvement in the performance of what you can extract from a single distant homologue being available for search models. And these just illustrate, like I say, the conserved catalytic site, which is automatically captured as by this truncation procedure. This is how you do it at the command line. It is relatively simple. There's two steps to Concord and 1 step to the normal modes method. I want to threw this in very much last minute, but so that's what we'd used Concord for before. But as we were looking, it was really quite educational. The cases in the MR clinic, some of you will have seen this over lunch, and thanks to Anna for allowing me to show this structure. So you might remember Anna's case had 2 domains, which were reasonably high sequence identity, but kind of awkward, and Rona managed to solve it by splitting into the 2 domains. When I ran this past the Concord approach, Concord generates structures where the 2 domains have different angles with respect to each other. These are different clusters, so these all derive from the same set of Concord structures, but you can perhaps see these are all superimposed on this lower domain here, and you can perhaps see that the angle of the upper domain varies quite a bit between these different clusters. This was the starting point for the analysis, and this was the cluster that ultimately solved, and this is the Shell-X trace of what came out unambiguously indicating success. So it's been a learning experience for us as well, realizing that Concord may be a route to search models that contain more than 1 domain, and you want to explore the domain angles between them. So finally, this big excitement in bioinformatics over evolutionary covariance, allowing us to predict these contacts, and what I'll be talking about today, making better models from as a result. We looked at this back in 2016, so the contact methods have actually improved significantly since then. But even at that point, we realized that factoring this information in allowed us to solve structures that were much bigger and much more beta-rich than we previously managed. So we struggled before with all beta proteins and anything much above 120. Here we extended the range considerably simply because we could build better models with the extra information from the covariance analysis. This is very simple to factor into AMPLE. It's simply giving it those contact predictions. I just want to mention here CONKIT, which is also part of CCP4, which can do a whole bunch of things related to covariance, but here might be useful for you in converting contact predictions from servers, which currently employ all sorts of different file formats, into the right kind of format that AMPLE wants. So all of the ab initio I've shown you so far has been fragment-based, but people have realized that with this new covariance-derived contact predictions, you don't need to build your models that way. You can use CNS, which older crystallographers in the audience will remember, and use the contacts as distance restraints and fold it up by distance geometry with additional constraints specifying secondary structure. One of the methods that works like that, so it's not fragment-based, it's much quicker, as a result is confold2. These are a bunch of targets where we modeled using Rosetta, confold2, and these other fragment-based approaches. They were not so good in our hands, at least. Confold2 turned out to produce models that were almost as good as Rosetta, occasionally better, but the results that we have so far, and admittedly the numbers are quite small, suggest that the confold2 models are not actually as good at solving structures by molecular replacement. So for some of these cases, the red indicates failure, orange means we just about solved it, green means we solved it. The white here means that we couldn't make models because in fact these were proteins that didn't have the covariance information required to fold them up with confold. So as things stand, we're still with Rosetta. We like the look of confold because the models look quite nice, but we speculate that they're not quite as perfect in their local geometry, and maybe that somehow impedes the molecular replacement. One of the interesting things about the contact predictions is you get almost two for the price of one when you're doing the modeling. They help build better models, but they can also help you select the better models, and that's also a challenge with ab initio modeling. And the reason for that is that the model quality, which is the TM score on the x-axis here, correlates reasonably well with how well the models satisfy the long-range contact predictions that you have. And so what we did was use that information to filter out, in several different ways, subsets of the models which we believe to be higher quality from this analysis here. And sure enough, we can start to solve some cases that we didn't solve before. So none here is the vanilla approach. These other three approaches are selecting within that pool of ab initio models based on how well they satisfy these long-distance constraints. And I just want to mention very briefly here, this is not related to the contacts, but Jens did a massive amount of benchmarking, and we realized that the initial way in which we'd set up AMPLE was not necessarily the most efficient for hard cases. So instead of hammering one single cluster, what we do now is sample more sparsely across 10 clusters, and that's better for hard cases, although if you actually know that you've got the right fold, then you may want to revert to the classic mode. And then finally, what we're quite excited about right now is the idea that you may not need to do any modeling at all. And the reason for that is that two of the main players in the field of modeling have done quite large-scale exercises in predicting structures for PFAM families that didn't have anything in the PDB. So both the Baker Group with their Gremlin software for contact prediction and obviously Rosetta, and the Pecons FAM people have produced databases or repositories. The Gremlin one is smaller because it only contains ones that they predicted were high quality. But interestingly for us, they present 30 different structures for each family. And that caught our eye, of course, because we like to work with a pool of structures and cluster within them and truncate down and produce ensemble search models. And so although we're going to be looking at this family on the right in the future, this is where we've focused so far. In the Gremlin, and these are some results so far. We've looked at targets where the models were first deposited and then someone else later came along and solved a structure. So these are genuine sort of blind, they're genuine sort of real-world scenarios. We looked at targets had up to 3 molecules in the asymmetric unit, a variety of sizes, identities, and resolutions. And we solved a few of them, which is quite nice, and not necessarily completely trivial ones. Sometimes we had to look in different clusters. Sometimes we had to truncate. I'll show you that on the next slide. This one was quite interesting to us because it's quite a large target, and in fact the model only represented part of it at 60% identity. So we've got a lot more to do to explore these, but we can already show that you can solve your structures using models that you just retrieve from a database and run through AMPL. And like I say, sometimes not just the clustering, but also that quite significant truncation is required to make them work. So this is the target that was solved, but only once you truncated down those original models and produced subsets which are actually much more accurate. I said I'd come back to the citations. So if you run AMPL through a GUI, there's now an extra tab here which not only lists all of the references that you should cite for all of the important underlying software, but also allows you to export them as a bibtex file. So there's really no excuse. It costs you nothing, and so if AMPL works, make sure you cite our AMPL paper, but also all of this underlying software, because that's important for the developers. So finishing off, when to consider AMPL. So AMPL is more for the hard cases, so if you've got a novel or divergent fold, especially if you've got good covariance information, it's worth thinking about. Works well on coils and works reasonably well on transmembrane helical proteins. These days, you might want to look in a database to see if there are already models of your target available, because the models are very good quality in that database. But also if you have one or more distant homologs that don't work easily, or an NMR structure. Now, I'll finish by just acknowledging the main people behind this. Jens is currently the main guy, and Adam's done a lot of the work recently that I've just shown you. Jacqueline produced it first, and Felix, although he's left now, sadly, he did a lot of the work too. Ronan's been a big part of this exercise, and Martin and Olga gave us some useful guidance, especially at the beginning. These are the institutions and the funders. And I'll just finish with this, is the page where you can get more details and the full documentation. And I'll take any questions.